That's a good one. Dr. Graham.
Yeah, the ones next to them. Yeah, 0 credit. I don't know. And then my college is 3, so I don't know. It must be a glitch.
You know what? I think it's because Dr. Graham if they operate.
Maybe that might not be.
I don't think they are the same to that. Yeah, I think you should do that as well.
Like it says 0 or 3 credits.
He's bad at a lot of the person. Oh. Okay, let's start again with a few practice questions. So this one I thought of a couple hours ago, and I think it's kind of challenging. Let's practice on hard questions, right? So read it carefully and whisper to somebody next to you what you think it is. All right, so when I'm making up questions for exams, it's very easy for me to kind of throw in a lot of words that kind of sounds like it might be right. So this was kind of what we ended, well, what we covered, one of the things we covered last time was thinking about what happens, how cytotoxic T cells are activated and what they do once they're activated. So when I'm thinking up questions, I usually try to throw one, if there's 5 possibilities, usually one of them is just silly. Like, okay, so the one here, all right, C, activated cytotoxic T cells that stimulate the production of antibodies. Whoops, oh, I'm on erase, that's why, okay. All right, so cytotoxic T cells don't make antibodies, right? Hopefully that we've got that ingrained, that it's B cells that make antibodies. So that one's not right. It's not B cells that stimulate cytotoxic T cells. They do, although make perforin and granzyme. That's how they act, but it's not due to B cell stimulation. B would be correct if I said MHC class 1. Okay, but I didn't, so that was wrong. Like I said, this one's. So then it comes down to these two, and I almost maybe thought I made a mistake when I read this over again. But the reason that A is not right is because cytotoxic T cells, they don't kill pathogens. Remember, they are killing our cells that are infected. Okay, so they are acting on host cells. So this is correct as well. Cytotoxic T cells, the response is going to be longer acting. It's going to be better if they also get cytokine stimulation from activated T helper cells. Okay, apparently they can act on their own, but it's a much longer acting, better, more efficient response when they're activated by the T helper cells. So, all right. Like I said, that one would have taken some parsing out during an exam. I think hopefully this one's a little bit more straightforward. We talked again about what superantigens are. Take a second to read that. So correct answer here would be B. E describes autoantigens. I just made the rest of the stuff up. So, okay, non-specific activation, a large number of T cells that cause release of lots of cytokines, overwhelming inflammation. So we kind of talked about that again last time. And then we talked about that terminology, active versus passive, natural versus artificial. See if you can apply those terms. So one more question. Okay, what do you think about that one? B, right. Right, okay, there's nothing natural about that, right? That involves technology. So, you know, involves syringes, an IV, or whatever. So artificially acquired, it's passive immunity because you're just giving the individual antibodies. The individual's not making their own antibodies. Okay, so it would be active immunity if the individual is making their own antibodies. Okay. Good. So picking up where we left off last time, we just began our discussion of vaccines, and we were talking about attenuated vaccines. In general, attenuated vaccines give us the best protection. Like they give a more natural immune response. Our cells will, you know, take up, you know, and just very naturally present antigen, lots of different epitopes. So we generally get the best protection from vaccines that are attenuated, live organism. There are huge drawbacks with it. And the drawbacks are such that if we have any other choice, if we have any other option, then we will take it and not use an attenuated vaccine. So we were just getting to the disadvantages. Whatever I scribbled there last time when I looked at that, I was like, that doesn't make any sense. I kind of got, my brain wasn't working at the very end of lecture last night. So when we finished, we were talking about contact immunity. So if you give somebody a attenuated vaccine such that they get mild disease, they can actually pass that on to somebody else. And then they contract the disease and in a mild form, but then they get immune from it. So that can be a good thing. But we, you know, in our society here, like that's going to be a bad thing. So a disadvantage is that you may have transmission of disease. We'll say, you know, of a typically a mild form. But we really don't want that because we don't want to, again, come in contact with somebody that's immunocompromised and would not have a mild form of the disease, might have a very serious form of the disease, or, you know, if we're around a baby or something, then typically we don't want that. In addition, it's a little bit more cumbersome to distribute because the stocks have to be refrigerated. If it's a live organism, a live bacteria, for example, you'd have to refrigerate it. So it might be harder to get to, add to the expense of getting it to a remote population of people. But really the biggest thing here is that there is the possibility, even though it's very rare, of a reversion to the infectious, whatever the infectious form of the disease. An example when that happened with the polio virus. So the Salk polio virus came out in about somewhere in the 1950s. That was the first vaccine that we came out with. That was actually a killed vaccine. We're going to talk about those next. In about 1960, the Sabin version of that came out. Again, Salk was a killed vaccine. The Sabin vaccine was an attenuated live vaccine. And in the United States, we switched to the Sabin vaccine because it was cheaper and it worked better. Okay, and that also you could give that orally. So it was one less needle poke for babies. You gave it in this little tube. A couple of my kids, I can remember, had that oral polio vaccine. Unfortunately, so we switched to that Sabin. Unfortunately, in the year 2000, they had a rare reversion event where that weakened strain of polio reverted back to the wild type. And about 20 kids in the Dominican Republic actually got full-blown polio from the vaccine. So even though the chance of that happening is really very remote, in this country, we don't use that Sabin vaccine anymore. So we've gone back to using the Salk one. And it works good enough because we don't have polio circulating here anymore. So we'll talk about that more. That Sabin vaccine still is used a little bit in other countries because it's so cheap. But eventually, hopefully, we won't have polio circulating anymore, and we won't have to administer the vaccine anymore. So some examples, though, of attenuated vaccine. The measles, mumps, rubella, that MMR vaccine. We have had other kinds of measles vaccines that they didn't protect well enough. So we have to give that in attenuated form. Some of the influenza, we have lots of different kinds of influenza vaccine. The one that works best in kids from about the age of 8 to 12 is actually a strain of flu virus that only replicates in colder temperatures. So it will replicate like in the nose, but not down in the lungs. Apparently that works best for, again, kids of a certain age. Chickenpox is another one that's attenuated. So still used. Probably the most common vaccines are considered killed, or if it's a virus, it'd be more correct to say inactivated, because is a virus really alive to begin with? Debatable, right? So they can either be whole cell vaccines or just a part of that pathogen. So if it's a subunit, we've usually taken some key protein that that elicits a strong immune response. So the COVID vaccines, all our vaccines are targeted toward the spike protein. That's the most antigenic part of that virus. So in comparison to the attenuated vaccines, an advantage, these are absolutely unable to replicate in the host. So if you've ever heard somebody say, oh, I got the flu vaccine and then I got the flu, okay, it was not from the vaccine. Okay, there's quality control in every batch to make sure that those viruses are inactivated. You cannot get the flu from the vaccine. Now, you could potentially get the flu at the same time from a strain that wasn't covered in the vaccine. That would be really bad luck. You might have, some side effects from the vaccine, but you didn't have the flu. In general, any kind of killed vaccine is you have to take more doses of it to kind of ramp up as good an immune response. And typically we're going to find mostly a humoral immune response. In order to get good cell-mediated immunity, our T cells have to kind of ingest the pathogen and present it with MHC class 1. And our cells just don't take up any kind of killed antigen as naturally as what it would a live pathogen. Typically, the antibody response is enough. Okay, so some examples: influenza, rabies, the Salk, polio, pertussis. So again, either that's a whole cell that's been killed. How do we kill or inactivate these pathogens? Either we hit them with some kind of radiation like gamma rays or UV rays, or we heat them up. Sometimes we treat them with a chemical like formalin, really small amounts, enough to inactivate them. One drawback with using the whole cell has come up with the pertussis vaccine. So we give pertussis in combination with diphtheria and tetanus. So DTP, that standard for diphtheria, tetanus, and the P was whole cell pertussis. The problem that we were finding with that, and again, this was a vaccine that we used, I don't know when we quit using this one, but the problem was the organism that causes pertussis, Bordetella pertussis, is a gram-negative organism. So you know what's in the outer membrane of gram-negatives, they are going to have endotoxin, that lipid A. And even though that outer membrane is going to be disrupted because the cell is dead, there's still going to be some endotoxin associated with that. And so patients would experience a good bit more of inflammation than you could argue what was acceptable. You know, when mothers vaccinate their babies and the baby's cranky for a couple days after that, it's going to make the mother less likely to go back for the next dose. So again, because it was whole cell, we'd experience a good bit of inflammation, mostly just, you know, the kinds of soreness and redness that typically is not a big deal. But to try to improve that, we've now switched to a subunit vaccine for pertussis. So again, a subunit, they usually just take a part of the pathogen that causes an immune response, maybe several different parts, and inoculate that. So it's less antigenic because there's just less epitopes that are presented with that. But we do have less inflammation. So now we switch to the acellular pertussis. So that vaccine is diphtheria, tetanus, and then the AP, DTaP, stands for acellular pertussis. So again, less inflammation, it tends to not work as well. Pertussis in particular, our immunity wanes usually by the time about 12 years of age. So babies that were vaccinated with the DTaP, you know, they don't have much protection by the time they're 12. And so they recommend that you get a booster when kids are about 12. And that just reminded me of one thing that I did right as a parent. I had my son vaccinated on his 12th birthday to get his pertussis booster, and he never let me forget that. Like, he was really mad that I made him get a shot on his birthday. But the following summer, he went away to summer camp for a week, and there was somebody in his cabin that came to camp with an active case of pertussis. Of course, I didn't know that at the time, and 2 other kids in his cabin got pertussis. But they had been vaccinated as babies, but then didn't have their booster, and my son had his booster. So yeah, I just felt like I did something right. Doesn't happen all that often. Okay, so again, a lot of our vaccines are some kind of killed vaccine. A couple of infections, it makes sense to, really the damage is done in our bodies due to some kind of exotoxin that's produced. So it makes sense to take that toxin, inactivate it, again, usually by heat or some kind of chemical treatment. And so that we call that a toxoid vaccine. It's just inactivated toxin. So that produces a pretty good antibody response. Really the two infections there, tetanus and diphtheria that are caused by the toxin. Again, big problem with this, there's not as many epitopes presented to our immune system. And so that's probably why immunity wanes. For tetanus, you need to get a booster every 8 to 10 years. You know, so usually, you know, that people typically kind of let that go a little bit. And then maybe if they're going to travel out of the country or something, they recommend, oh, you know, get a tetanus before you go in case you don't have access to medical care when you're in that another country. It's pretty rare, tetanus, especially here, but other places, those spores, it's a spore former, a Clostridium, might be more likely to get that in a cut, for instance. Diphtheria. This is an infection that we won't talk about anymore. Before we had the diphtheria toxoid vaccine that was formulated and came out in the 1920s, we had over 200,000 cases annually in the US of diphtheria, 10 to 15,000 deaths a year. Mostly children. That infection causes this kind of pseudomembrane that forms in the throat and can actually suffocate someone, lots of other complications associated with it. So now that people, you know, most babies get vaccinated, we have less than 1 case a year in the US. So we've really come a long way in tamping that infection down. Okay, so there are some other kinds of vaccines that are used. A newer one is called a conjugate vaccine. And really, you can think of this as kind of a subunit vaccine. Sometimes you might call it a subunit vaccine. But this is where, for several different infections, we've taken, remember that polysaccharides that make up the bacterial capsule. And that typically is not terribly antigenic. So we've conjugated that or attached it to a carrier protein that is more likely to be noticed and will give us a better immune response to those polysaccharides. So again, this is especially important for those polysaccharides that are T-independent antigens, because kids especially have a very poor T-independent antigen response, very, very poor B cell activation to those antigens. Okay, so some examples of this, the strep pneumoniae vaccine, we'll talk, whoops, pneumoniae, whoops, AE, oh, that didn't look right. That vaccine is called Prevnar, it's actually Prevnar 23, old picture there. So they've taken 23 different variations of the capsule to that strep pneumoniae and incorporated it into the vaccine. So that's, again, we keep making that one better and better. It used to be only 13 different variations of it. Now there's 23. So that vaccine, especially given to young children, the whole different formulation for older adults to try to protect them from pneumonia. Here's another organism that we'll be talking about soon, Neisseria meningitidis. Another tough one to spell, but that's causes a bacterial meningitis. Sometimes we call that the meningococcal vaccine. Again, polysaccharide, capsular polysaccharides conjugated with a protein. Another one we mentioned already, Haemophilus influenzae. that's that Hib vaccine, Haemophilus influenza B. I mentioned that before as just an example of a T-independent antigen. Okay. And then I would be remiss if we didn't talk about some mRNA vaccines. Of course, the Pfizer and the Moderna COVID-19 vaccines. This is technology that this company Moderna modify RNA. That company's been around a while. The technology has been in the works for 30 years or so. Originally, they were trying to use RNA as a drug to target certain kinds of diseases. And they found that they are more successful in making it as a vaccine, but it does have really, it's a lot of exciting potential around using RNA to treat lots of different infections and to treat cancer as well. So it's kind of exciting technology. The big advantage is that it's really quite safe in terms of there's no chance of actually it causing the infection that it's trying to prevent. There's no chance of any kind of mutation, again, in that RNA, or it can't mutate the host either. And production is really quite quick and inexpensive. If I remember right, in 2020, when they came out, the early spring, when they were just kind of figuring things out in March there, they came out with the sequence of that coronavirus. And within a couple of days, they had made an RNA vaccine against it. Now, there was, the argument that we should have started distributing that right away, but we had to go through the protocols and test it. We'll talk about how we test vaccines, make sure that they're safe. And of course, that took months to make sure that it was safe, but we had the ability, we had the technology to come out with it right away. Of course, there's disadvantages. Messenger RNA degrades quite quickly. So in terms of, keeping the stocks, so we have to keep it at very, very cold temps. And that's just so the messenger RNA doesn't degrade. There was hurdles to overcome so that the messenger RNA didn't degrade too quickly as it was entering our cell and as it got into the cell. There's lots of kind of nucleases that would degrade it. Short-term inflammatory reactions are maybe a little bit more common for this type of vaccine than they are for others. We have toll-like receptors that will recognize types of nucleic acid that aren't our own. Okay, so we talked about macrophages having toll-like receptors that recognize PAMPs. Okay, an RNA from a virus, it would be detected as a PAMP. So there were hurdles to try to prevent RNA immunity from having too much of a reaction there. It's very, very effective, especially at preventing severe COVID-19. The one side effect that you hear of is myocarditis, inflammation of the heart muscle. Much more prevalent in boys and men, young men especially, for some reason. Not sure why. It usually just manifests as a little chest pain and it goes away. And it's pretty 1 in 50,000. They've discovered that, of course, when the vaccines just rolled out, we wanted to get everybody have both, the vaccine and the booster as close in time as possible so that people could be protected as soon as possible. They found if they wait longer for that second dose, maybe wait 2 months instead of one month, and they think the incidence of the myocarditis goes way down. And I should add that if somebody gets COVID, then the chances of them developing myocarditis are way higher than if, from the shot. So you always have to weigh the risks, right? The DNA vaccine that came out was a Johnson & Johnson vaccine to COVID. Sometimes we call this a viral vector vaccine instead of a DNA vaccine. It's both, really. What was new about this vaccine that it utilized a virus called adenovirus that usually infects chimpanzees. It gives them a mild cold. So it was really a clever way to use that virus to deliver this piece of DNA that encoded for the spike protein. So I forgot to say that about the messenger RNA, that messenger RNA encodes for the spike protein and gets our cells to actually present that spike protein, and that elicits an immune response. So this is a virus that can't replicate in humans, so that's very safe. DNA is a little bit more stable than RNA, so there was they don't have to keep it quite as cold. And it seemed pretty effective with one dose. I think that they did start to give booster shots to that as well, just so it'd be even more effective. Today, we are not using the Johnson & Johnson shot unless for some reason somebody can't have another type of COVID vaccine, because it does have the very rare but serious side effect of this clotting disorder called thrombosis with thrombocytopenia syndrome. How rare was it? Let's see, I looked that up. Looks like 3 in a million. So very rare. I mean, that's rare enough that they're not totally sure that it's from the vaccine. But some people have died from this. So for that reason, we're not using this vaccine again, unless we don't have other options. This Guillain-Barré is a infection that sometimes people can get from any viral infection. It's neurological, so it's a little bit scary. It can cause paralysis. Usually, lasts, a short period of time, and then patients recover, but that has potential to be dangerous. Let's see, the incidence of that about 1 in 120,000. So again, weigh the risks. One other type of vaccine that I want to mention, because we did talk about malaria earlier in the course, and this is a very new vaccine. It's called a recombinant vaccine, and it's a vaccine to malaria. So it has the trade name Mosquirix, a little mosquito in there, that makes sense. And the other name for it is RTSS. Which is not intuitive for anything, but I'll talk about where that name came from in a minute. Okay, so we call this a recombinant vaccine because what they've done is used molecular biology techniques to kind of piece together different pieces of DNA that encode for certain antigens and kind of piece that all together. So they've used, remember the causative agent of malaria, the most severe kind of malaria is Plasmodium falciparum. And they've taken a protein from the surface, just to kind of remind you, I didn't post this, but we talked about this a long time ago. When malaria infects humans, first place that it goes is, I think it's a hepatic vein, so one of the veins in the liver. Okay, so malaria, super different stages, really complicated. They name all these stages something different, but this stage that infects the liver is called the circumsporocyte stage. So they've taken a protein from that stage on the outside of Plasmodium, and they've also taken surface antigens from hepatitis B virus. Why have they brought hepatitis B virus in this? Well, we actually have a vaccine, a conjugate vaccine to hepatitis B virus. So they've taken the technology that they use there, and they've just used that hepatitis B antigen to elicit a stronger immune response. So this name, RTSS, again, this was something that I didn't post, but I was just looking at this a little bit more carefully this morning, so I threw it up here. So that R stands for a repeat region of the gene, a repeat region of the gene that encodes for that CSP, that circumsporocyte protein of Plasmodium. And then they've taken another gene of that CSP, the T. So this stands for the R, the repeat. The T is these T cell epitopes of that same protein. And then the S is this surface protein of hepatitis B. Okay, so RTS, and then some more S, some more of this surface protein that apparently will kind of glob onto it. So again, that's just, I like to know where names come from, and then that kind of helps me understand if I see RTS again, what that means. Okay, so they've used molecular biology techniques. They've made this gene, they've inserted it into either bacteria or yeast, and then the yeast or bacteria will produce that protein for us, and then they use the protein in the vaccine. So apparently, we need an organism to make, ramp up and make a whole bunch of it, and that's a lot easier. So enough of how they make the vaccine. For years, when I taught this course, I said, oh no, we don't have a malaria vaccine. There's too many different variations of the surface proteins on the Plasmodium. It's just not possible. Well, it turns out they started they came out with the first variation of this vaccine in 1987. So this has been a long time in the making. They've been probably tweaking it all that time. What it does, it induces high enough antibody titers so that the parasite can't infect the liver. Okay. A huge drawback is that it takes 4 injections, and right now they're just giving it to kids because kids are at the highest risk of developing severe, life-threatening malaria. So, in remember, it's sub-Saharan Africa that has the most problem with malaria, and it's, populations that's hard to get to. So that takes some organization to get 4 injections, and it's only about 30% effective. So usually a vaccine is not going to be approved if it's only 30% effective. But in this case, because malaria kills somewhere between 1,000 and 1,200 kids every day, if we can save 30% of those kids, then it seems like a worthwhile endeavor. So it's been given, this was approved just recently, last year, 2019. And so, it's been administered to over 800,000 kids since 2019. So it's kind of gaining. Hopefully, again, they're calling it the first generation malaria vaccine. So they're hoping to improve on it, make it even more effective, and save a lot of lives. It's the only vaccine that we have to a protozoal or a parasitic infection. All right, so a couple last comments about vaccines. Those are all the vaccines that are kind of FDA approved right now, all the different kinds of vaccines that are out there. Of course, it came up the last couple years. Did we roll out the COVID vaccines too quick? People didn't trust them. So it's reasonable to kind of understand the kind of testing that has to be done before a vaccine is approved and considered safe. Even before we start phases of clinical trials, and they talk about this up here, we do a lot of testing in animals with a vaccine that's made. Sometimes we don't have a good animal model for the infection, but at least we can see if the vaccine has any adverse effects in the animals. Okay, so once it seems safe in animals, then in phase 1 clinical trials, they'll give it to a really small number of otherwise healthy people, and they'll monitor those people really closely to see if they have any side effects. Hopefully, maybe try to understand if it's actually effective. And then if everything seems all right, they'll move on to phase 2 to give it to maybe several hundred individuals of varying health status. So not necessarily healthy people. And then again, maybe play with the dosage a little bit. And of course, you're giving a placebo to a group of people that you're watching very closely as well. That's important. Everything seems fine, then you're going to start giving it to thousands. I think with the Pfizer vaccine, they give it to 30,000 people. Again, monitored them, gave the placebo to people, and they had to watch for any adverse effects for 2 months after that. So with any other vaccine rollout, any adverse effects were always apparent within the first couple weeks. Okay, so monitor them closely. At that point, if everything looks good, then the FDA would approve. If it meets standards of effectiveness, standards of safety, an independent advisory team will approve it. So it's not the drug manufacturer or the vaccine manufacturer that's approving it. It's supposed to be independent people. And even after that, there's what you could consider a phase 4. So after it's been approved and it's given to the general public, then it's called the Vaccine Advisory. It's VAERS. Vaccine. Oh, sorry. Vaccine Adverse Effects. I'm blanking on what vaccine adverse effects reporting system. Okay, so the VAER, the VARS. Okay, so traditionally, if say a baby had an adverse effect, the parents would take him back to the doctor, they'd call the doctor to report it. I know that when I got my COVID vaccine, they asked me if I wanted to opt in to getting texts from VARS. So I would get text, maybe the first few days and then every week for a number of weeks. And after I got my boosters as well, asking me, how are you feeling today? Any change since, you last had your vaccine? So just so they have as much data as they can, just to know if things are generally safe. Okay. So, in general, no matter how you claim down on COVID, weighing the risks, whether you got that vaccine or not, you really can't argue with the data that vaccines have incredibly made a huge difference in the number of infectious disease we have. If you take, just a couple, here's polio, here's measles. If you look at the incidence, remember incidence is the number of new cases of a disease. Then, polio in the 1950s, something about like 40,000 cases a year. We had a couple different vaccines that came out. Now we only have, wild polio in 2 different countries, Pakistan and Afghanistan. So we're hoping to eradicate that totally. There's no animal reservoirs or anything of that. So we're hoping to totally eradicate that. You might have heard in the news this fall or last summer, there was somebody in New York City that had polio. And I read a little bit about that. They think they could sequence, the virus that he was infected with. And they think that he got that. It was actually, the virus was derived from the Sabin polio vaccine. So they still give that attenuated vaccine in other countries. And when people get that vaccine, they shed polio virus in their feces. And, so it can kind of be maintained in waterways, and it mutated a little bit. And especially in places where their sanitation is not good, he somehow contracted that. And then in another country, again, it was another instance of reversion, and that showed up in New York City. So again, hopefully, eventually we won't have to give any kind of vaccine, and there won't be any chance of that coming back. Measles, Thanksgiving is coming up, ask your grandparents if they remember having measles as a child, because almost everybody had it. And there was a lot of complications associated from that. So our first measles vaccine didn't really work that well. When the form that we use now came out, I think it was the late 60s that it came out. We had this kind of bump here in the late 80s of measles, and that's when they decided, okay, instead of people getting one dose when they're 12 months or 15 months and another dose before they start school, they need a booster like before they go to college. And then that catches like the 5% of people that didn't have total immunity from 2 doses of it. So now that we give kind of 3 shots of that, again, really rare, but we have to keep the vaccination rate for that high, or else we start seeing outbreaks of that again. Okay, so any questions, anything about vaccines that you wonder about? Okay, that took longer than I thought today. That's okay. I didn't really, I was gonna, of course, we have an exam a week from today. I think probably that exam will go through Staphylococcus. So I don't have a lot of slides talking about staph. We'll finish that up on Monday. And so I'll get something out about that on Blackboard. Just to kind of introduce this, we have a few minutes left today. This is kind of the part of the semester that we like to call our bug parade. So we talk about lots of different specific infections here at the end of the semester. So very, very clinical focus. How I have always taught this, I've just talked about certain pathogens, starting with the gram-positive bacteria, and talking about acid-fast organisms, some gram-negatives, and then talk about some viral infections. OpenStack's textbook comes at it from a different angle. If you've looked ahead, they talk about skin infections and respiratory tract infections and gastrological infections. So I didn't have time to change all of the way I taught it to mimic that. So it's a little bit different. You might have to hunt around a little bit for information. I'm trying to tailor the homework questions so that they're only asking about things that we talk about in class, and you don't have to go through it's great to read the textbook, but again, it's going to throw in a lot of different organisms instead of just kind of focusing on one all at one place. Okay, so we will, I think we'll get through staph pretty easily on Monday, maybe start streptococcus, but I think we'll just include staph on the exam on Friday. And that seems like a good place to stop. So have a good Friday. Bundle up, stay warm this weekend.